How are you doing, Jack?
Doing well, thank you. How's everything going there?
Good. It's starting to get a little bit warmer. Almost feels like spring here. Jack, what
Excellent.
would you have been doing if you didn't choose science?
I do if I didn't choose science. That's a hard one. I don't know, is the honest answer.
So you would choose science.
Absolutely, yes.
Okay, we'll get started. Our next speaker coming from the UK is Jack Turner, and he'll be telling you a little bit about EMDB Empire. Take it away, Jack.
Cheers, thank you. Hi everyone, I'm Jack. I work at the Electron Microscopy Data Bank, and today I'm going to be talking to you about the archiving of structural biology data. Hopefully I'm coming through okay, and hopefully you can see the red cursor here. So just before I get into the talk, I just want to acknowledge the people that do the work behind the scenes. So first things first, the EMDB team, if it works. There we go. So the EMDB team is currently made up of 8 members, if I include myself. Our most recent addition is Kyle, who is the new EMDB team leader. And everyone on this page has been somehow involved in a lot of the work I'm going to talk about today. But the collaboration extends far beyond the EMDB team. And when we're talking about the archiving of the type of data we're talking about today, we have to acknowledge the Worldwide Protein Data Bank Consortium, of which the EMDB is a core member, but it also includes RCSB PDB, PDBE, PDBJ, and BMRB. And very recently, PDB China also became a member of this team. So recent, in fact, they don't have their own logo, so they have PowerPoint stock font for the time being. And then outside the Worldwide Protein Data Bank, there's the Empire team as well. And we work very closely with them. So whilst they don't come under this umbrella, they are still very much closely linked to the work we do. In terms of an overview of today's session, I'm going to start just by introducing the structural biology archives. I'm going to spend a very short amount of time introducing a few features of the EMDB website, just to highlight those to you. And then I'm going to spend the latter half of this talk talking about depositing data. I've chosen to focus really on this as the main thing, both because it's a bit hidden. So if you haven't previously deposited structural biology data, you probably won't have seen this. But also, if you have previously deposited data to us, you'll know it can be quite painful. So I'm going to give you some tips and tricks on how to make your life a little bit easier in this regard. So let's start with introducing structural biology archives. But just before we get into that in full, I like to start with this figure here, where we just have all the scales of structural biology across the top here. And then we have a bunch of horizontal bars, each with a methodology associated with them, roughly showing the scales that they collect across. It's not entirely accurate, but it gives a rough picture. And the reason I like to start with this figure here is because we're here to talk about electron microscopy and the archiving of that type of data, but you're all structural biologists, you're all working on biological systems. You may end up going and using other imaging techniques and things like this during your careers when you're asking particular biological questions. And I just want to highlight that there is always an archive that this data can go to. So X-ray crystallography, this can go to the PDB. Cryo-EM data and cellular electron microscopy data can go to EMDB and Empire. Small angle X-ray scattering, this goes to the SAS BBB. All of these photon-based techniques shown in green, they can all go straight to the BioImage archive. And then finally, volume scanning EM, but also X-ray tomography techniques, they can go directly to Empire. And I'll talk a little bit more about how that works in a minute. So yeah, just to highlight that there are a lot of structural biology archives out there that you can deposit to. So today, although we're focusing on EMDB and Empire, and we'll talk a bit about the PDB as well, because we can't really talk about those without including the protein data bank. So just to start with, an introduction to the Electron Microscopy Data Bank. It was established back in 2002 at EMBL-EBI, where it's still located today. It became a WWPDB partner in 2021, so I briefly talked about this umbrella organization earlier. And it's the home to electron microscopy volume maps and tomograms. All the data is freely and openly available under a CCO license. I have this bullet point on every single one of my slides where I introduce an archive. What this really means is you can freely download and reuse the data in any way you wish. A couple of examples of the type of data you might find in the archive are shown here. And then on the right-hand side here, I'm just showing the current holdings of the archive and the growth over time. And the reason I like showing this is because it's quite indicative of the field as a whole, where we are seeing exponential growth in the total number of entries in the archive over time. And this trend seems to be continuing. And if we look at the number of entries released annually, this is growing linearly as well. The final point is lower because I took this just 1 month into 2024, and this final data point is 2024. So that will catch up with everything else. Something else I want to highlight is there is database publications. So we do publish occasionally what we've been doing and our future plans. And our most recent publication went out at the end of last year and was in the January issue of NAR this year. And one of the figures I made for that paper is this one here. And just to highlight, considering this exponential growth, so here we're looking at the number of releases of X-ray entries shown in blue with a trend line in light blue, and then the total number of EMDB releases and the total number of EMDB releases with a PDB model shown in green and orange respectively, with this dashed line showing 2022, and then the trend lines extrapolating beyond that. There's often a question of where we're doing in comparison to the other most popular structural biology method, which is X-ray crystallography. And if this trend continues, we are looking to reach parity with them in terms of the total number of yearly releases in sort of 2025, 2026. So that's fast approaching. So that's the Electron Microscopy Databank. Now let's move on to the Electron Microscopy Public Image Archive, or EMPIAR for short. This is the home to the raw imaging data used to produce the maps found in EMDB, but its scope has since increased to also include volume EM techniques and X-ray tomography and microscopy techniques. Once again, all that data is freely and openly available. And a couple of examples of the type of data you might find are just shown here. So this one on the left has an entry in EMPIAR where all the raw data is, but it also has a high resolution structure in the EMDB. So if you're interested in learning reconstruction methods, in theory, you could go now and download this EMPIAR dataset and try and reproduce this structure in the EMDB. It also has volume EM data. So here's a volume EM tomogram of Plataneris damarili. Hopefully I'm pronouncing that right. It's a worm, essentially, but just to show you the various things that we have in the archive. Let's change that to a laser pointer. In terms of the holdings in EMPIAR, the numbers are much smaller. So we have over 33,000 entries in the EMDB at the moment. We have just over 1,500 entries in EMPIAR. This is because it's not mandatory to deposit to EMPIAR. It's just highly recommended. But that's not really the whole story. When it comes to EMPIAR, it's more about the total data that's accessible to the community. And actually, if I just change this graph very slightly to change the axis to the total amount of data available, you can see we're now serving over 4 petabytes of total data to the community. So this is a huge amount of data. Last year, I was coming out and saying we've reached this incredible milestone of 3 petabytes, but actually, it really didn't take us long to get to 4. And you can see last year, we added 1.5 petabytes worth of data to the archive. So really incredible numbers here of data that's freely available. So that's EMPIAR and the EMDB, but we can't talk about those two archives without talking about the Protein Data Bank as well. And the Protein Data Bank is the archive for coordinate models from EM, X-ray, and NMR techniques. In terms of the total holdings, here I'm just showing a graph which shows the total number of EMDB entries with a PDB model fitted in. So currently, or as of when I took this graph, there are about 18,500, about 33,000 entries in the EMDB. So about 55% of those have a fitted PDB model. And the Protein Data Bank is a bit interesting in terms of how it's managed, in that it's a collaboration between 3 sites. So PDBE, RCSB, PDB, and PDBJ. And they all collaborate at the point of data in, and they all work very closely together to develop these pipelines to make the user experience better for you guys as end users. But they all compete at the point of data out, and they all have their own websites. So they're shown at the bottom here. Each of these websites does serve exactly the same coordinate data. So the underlying coordinate models are all identical on each of these websites, but the way that they display that data, the things they do to sort of add features to that data is different. So it might be worth going to all of these websites and figuring out which one works best for you. And they're constantly adding new features because they're in constant competition. So this works well for you as end users as well. So just to summarize the different structural biology archives, taken together, they allow the archiving of the full electron microscopy processing workflow. So EMPIAR, all of that raw data can go there. The EMDB, the refined electron microscopy map that's in your publication, that goes there. And if you have built in a model, that will go to the Protein Data Bank. There is one other archive, though, that I want to quickly talk about, and mainly because the software behind it has become increasingly popular, and that's AlphaFold DB. So AlphaFold DB is a collaboration between EBI and Google DeepMind, and it currently contains over 200 million protein structure predictions. To give you some context, PDB contains just over 200,000 structures. These aren't completely comparable because AlphaFold works on single chains, whereas PDB is multi-chain. But PDB also has multiple models of the same protein. So actually, if I were to say that AlphaFold DB contains 1,000 more proteins, that wouldn't be probably too far off the mark. The reason that I want to mention this is because whilst AlphaFold has been really, really good for accelerating science, it was kind of the first form of generative AI that got given to the masses to use. And it became evident quite quickly that by EBI and Google DeepMind, that they wanted to create educational material to make sure that people were empowered to use this type of technology responsibly in their work and validate the structures that they were creating. And something that came out literally just a couple of weeks ago was the first one of these educational courses, which is accessible here. I will put this QR code up at the end if you're interested in going and having a look. It's a 3-hour-long course. It's completely free to access, and you can do it in your own time. And it will go through how best to use the database and AlphaFold and how to validate the structures that you produce using this generative AI method. So I think this is highly worth doing if you are considering using AlphaFold in your work. Okay, so that covers everything I wanted to say in terms of just introducing the archives. Now I'm going to very, very briefly go over the EMDB website and highlight a few features there. And then we'll get on to deposition and how deposition is carried out. So there are 3 main things I want to talk to you about in terms of the EMDB website, and that's EMDB search, EM validation, EM statistics. On the side of EMDB search, I just want to highlight that it's a bit more complicated than it might initially seem. So in this example here, I've typed in the word ribosome, and if I clicked enter, it would return all of the ribosome entries in the archive. But we have another system working in the background in the EMDB website called Emix. And what Emix does is for every single entry which is released, it goes and data mines 21 other resources and pulls in various annotations to allow you to analyze and group the biological data in different and interesting ways. And one thing that happens when you start to type something in is you get a bunch of autocomplete options. If you're on a widescreen computer, this will be like 20 plus boxes. I've just minimized this into 4 little boxes here, so it's easier to visualize. And some of these are the types of things you'll expect. So I've typed in ribosome. The autocomplete options are things like 70S ribosome, titles which include the word ribosome, so on and so forth. One of these options, though, is one of those 21 extra annotations, and that's the gene ontology section. And this allows you to group entries in new ways, so you can actually search for proteins involved in ribosome biogenesis or proteins involved in ribosome binding. This works for all types of gene ontology terms. So if you're interested in, say, viral membrane proteins, you could type in viral membrane proteins and you could group the proteins in that way as well. So just worth noting that there are interesting ways to group these proteins, download metadata, and do interesting statistics on this. Another thing to highlight in the bottom here, I've got an advanced search query. In this case, I'm searching for single particle entries. And then another thing that we data mine is the AlphaFold database. So these single particle entries must have a link to the AlphaFold database, but then they do not have a link to fitted models, where fitted models in this case would be models in the protein data bank. So this will return all entries where there is only an EM map, but there is actually a model in the AlphaFold DB, even though there isn't one that was originally deposited with data. And I think last time I did this, there were over 1,000 entries which have this kind of information. So you can see how powerful that could be. So that's all I want to say in terms of EMDB search. The next thing I want to talk to you about is EM validation. And I want to talk to you about this more generally in terms of the WWPDB. Anything that is released on the EMDB or Protein Data Bank website, we will not stop the release of data just because the data itself is bad according to the validation metrics. We have a bunch of policies and procedures that depositors must follow, but if the data validation metrics themselves are bad, we are not the structural biology police. We do not stop the release of that data. We provide validation reports that reviewers should read to assess the quality of the structural biology data. And then when the data is published and we release it, we provide all of these validation metrics to the community, freely accessible, so that you as the community can assess the validity of the structures you're looking at. The reason I highlight this is because it's very easy to go to the Protein Data Bank and download a coordinate model and use it in your work. But if you do that and it is an EM model, please come to the EMDB website and have a look at all of the structural biology validation metrics that we've got on the EMDB website. I'm just showing a couple of examples here. The classic one, the FSC curve, shown in the top right. We also have things like Q-score mapped to the model, which I'm showing here. We have a whole bunch of other metrics like RAPS plots, orthogonal projections, And we've got a whole bunch more coming as well, like the phase randomized FSC, map model FSC, things like this. Something we're also working on, because we're very aware that there are a lot of validation metrics in the world of electron microscopy right now, and it can be a little bit overwhelming, and we do present a lot of those metrics on our website, which can also be very overwhelming, is to try and make summary tables so you can assess the quality of an entry at a glance. And that's what these sliders are at the bottom here. So in this case, we've got 4 different metrics: Q-score, which is also shown here, atom inclusion, CCC, and SMOC. And we've got these diamonds that basically tell you how good this entry is relative to the rest of the archive. And we're working on bringing these to production soon, and hopefully these will make their way to the WWPDB validation report, so it'll be easier to understand at a glance. It's worth noting that these are just map model validation metrics at the minute. These don't validate the map. We are working on ways that we might be able to include map-only validation metrics in these sliders as well. And then the final thing I want to mention is EM statistics. So this is the EMDB homepage here at the top, and we have this big yellow button. And if you click that, you get to a web page with dozens and dozens of graphs that show the EMDB archive in a number of different ways. This one here is my favorite, which is EMDB entry resolution in shells per year, where you can actually plot the resolution revolution. You can see it happening over time. And we have a whole bunch of these graphs. And I just want to highlight this here, because if you are giving talks and you want to give a high-level overview of the field early in your talk, this is quite a nice place to come and get this type of information. And we do have a bunch of those that you could just download. Okay, so that covers everything I want to say in terms of the EMDB website. Let's spend some time now talking about deposition. And I'm going to do this in 2 parts. So we've got EMDB and PDB deposition first. That uses the same software package, so I'm going to explain how all of that works. It's also the most complicated of the deposition processes because there is a lot of things like metadata to provide. I'm then going to, after this, show a few slides on EMPIAR deposition. This is a lot easier in many ways and doesn't have quite as much data you need to provide. I should also note I'm not an EMPIAR employee, so I've worked with the EMPIAR team to produce these slides, which I hope will be helpful to you. But EMDB and PDB deposition is what I'm involved in directly as well. So in terms of the deposition process, we can't talk about this deposition process without mentioning OneDep, which is the software which is used for the deposition. So it's a unified deposition system for EM, X-ray, and NMR methodologies. So they all go through this one system, and it's hosted at 3 sites around the globe. So we have RCSB, PDB in the US, we have PDB in EMDB, and we're co-located in Cambridge in the UK, and then we have PDBJ in Japan. PDBC recently came online, and they will get their own servers at some point, but currently their servers are also hosted in Japan. And depending on where you are in the globe, depends on where your data will go for the process of annotation. So whilst we at the EMDB are located in the UK, if you're depositing EM data in the US, that will go to RCSB for the process of annotation, and you will talk to the annotators there. The idea behind this is that we geographically distribute the annotation workload, and it also means you as end users, you are constantly communicating with annotators that are close to you in terms of time zone, so you should get reasonably rapid replies. What happens though at the time of release is where we take responsibility. So the EMDB in the WWPDB consortium is what's known as a core archive keeper. So if you have deposited EM data in the US to RCSB, at time of release, it will be synced across to EMDB, and we're in charge of doing things like backing it up and serving it publicly. Conversely, the coordinate models, PDB, RCSB, PDB is the core archive holder of coordinate models. So if you deposited coordinate models in the UK, they will be synced across to RCSB at time of release, and RCSB is in charge of backing all that up and serving it publicly and all of that good stuff. PDB and PDBJ are also core members, and so they get access to the data early. So those websites I talked about before, when it comes time for release, they all have exactly the same data. So this is how the data is deposited around the globe. In terms of the type of data you can provide, the EMDB accepts 5 different methodologies, so single particle, subtomogram averaging, helical reconstruction, tomography, and electron crystallography. This doesn't quite tell the whole story though. There are a number of different deposition types that you can do. So you could do a map-only deposition, a model-only deposition, or a map model deposition. A map-only deposition, the classic example of this would be a tomogram, but there's nothing to stop you doing anything else as well. So in this case, this entry is quite a high resolution structure, but it was deposited as map-only deposition. A model-only deposition, you can do this, but it's worth noting that any model in the protein data bank has to be supported by primary experimental data. So the reason this facility really exists is if you previously deposited a map-only deposition, and for any reason later on you want to add a model to it, you can do this by doing a model-only deposition, and then you don't have to worry about resupplying all of those maps and that map metadata. You can just add a model to it. And then the map model deposition, that can all be done at once and it all gets sorted out for you. In terms of where the data goes at the end, you don't have to worry about that. The one-dep system knows that the models go to our CSV and PDB and the maps come to us, and it will sort that all out for you. I'm now going to show you a short video on the deposition process. I don't want to spend too long on this because it's mainly just a web form. I'm just going to highlight a few things as we go through. Mainly, this is for people that haven't seen the deposition process before. Those that have, though, I'm hoping I'm going to highlight a few things that may make your life easier in the future, particularly if you haven't deposited in a while. We have a few new features. So the first thing I want to highlight before I start the video is you could previously just start a new deposition and you get emailed a 10-digit deposition ID, and you'd have to keep all of those deposition IDs in a logbook and remember what on earth was going on with all of them. We've recently added this sign in with ORCID button, so I'll start playing the video now. Please do use that because that will associate all the depositions with you and you'll be able to deal with them all on the website. So the first thing I'm going to do here is sign in with ORCID, and you'll see that when I do this, it will recognize that it's me and it will tell me that on the one-dep website. So here we go, I'm signed in with ORCID here. And on this test server, I don't have any depositions, but if I did, they would all appear in this table here. You can see 10 per page of deposition ID, title, so on and so forth. So it just keeps all of your depositions in one place, and we didn't previously have this functionality. So I just wanted to really highlight that there. You're then going to start a new deposition. So in this case, I just have to choose country, fill out some basic information, and this is where you're going to set up the type of deposition you're doing. So in this case, I've chosen single particle. I'm saying that I'm not going to deposit any coordinates with this submission, so it's going to be a map only deposition, and I'm leaving this is it a composite map deposition as no. For the purposes of this talk, because of time constraints, I haven't gone into composite map depositions at all, but if you are depositing composite maps, please go and look at section 1.3 of the EMDB policy, and that will run through with you how to deposit a composite map set of entries. You'll have to do multiple depositions and you'll have to deposit the composite map, focused refinements and consensus maps separately. If you are at all confused about what you have to do, please don't hesitate to contact us. We can help you out. We're also working on a guide at the minute to basically show the easiest way to do this, because we are aware that it's quite involved to be able to carry this out. Okay, so you just have to prove you're human by typing in this number, start deposition, it will wear away for a second and you'll be taken to an instructions page. You'll then get to the file upload page. And on this page here, the first time that you log in and the first time you do file upload, you get this option here based on previous WWPDB deposition. What this lets you do is pull metadata across from previous depositions. Just for context, the EMDB schema contains about 500 metadata items that you can provide. That is split across 5 different methodologies, but you can understand this is a lot of data that you need to provide. So to make things easier, we do have a couple of methods for auto-filling the deposition interface. So if you're depositing multiple structures and you know things like your citation information, your authors, and your metadata are going to be very similar, you can provide that information here and tick these boxes and pull that information across. In this case, I'm not going to do that. I'm going to use another method for automatically filling the deposition interface, which is to use the MMSIF file format. And I will talk to you a bit more about that after this video. So here, I'm just going to go through, find the data. You won't have to watch this for too long. It will skip ahead in a minute. So here, just uploading a CIF file, which is going to contain a lot of that metadata, a map file, so on and so forth. And then when you've uploaded everything, it looks something like this. You're going to have to select the file type, provide some metadata on each of these files. So I don't really know these maps. I'm just saying the pixel size is 1 and the contour level is a very low number so that it doesn't error on me. And this will also skip ahead in a second to fully filled out. So here we go. Primary map is given the data type EM map. Half maps are given the data type half maps. I've got a metadata CIF here, which is given the metadata MMSIF, and I'll talk to you about how to make one of those soon. Just to highlight, the entry image must be an image of the entire primary map. We do now give you the ability to review this as well. Previously, you didn't have that ability, so people would accidentally upload pictures of their cats, but you do now have that on the website here, and you must make sure it is an image of your primary map. You'll then click Continue Deposition. This will whir away, sometimes for like 10 to 20 minutes. This is going to skip ahead, but it's basically doing lots of processing in the background, and then you'll get access to the full deposition interface. And basically, this is just a web form that you have to fill out. So as I go through this here, you'll quickly see that it's fairly basic information. I'm skipping ahead on some of this, so you don't have to watch me fill everything out, and you just go through this essentially. Something that I do just quickly want to highlight, though, is each of these pages is an individual page that you have to completely fill out. But you'll see a lot of these already have green ticks. That means all that information that's required is already there. And that's because I embedded all of that information in this MMSIF file that I was talking about. And even in the macromolecules page, where you've got this red exclamation mark because you still need to provide more data, you can see that actually as we scroll through this page, there is stuff that needs to be provided, but a lot of the information is already there. So it's all filled out. Another example of the type of data you can provide is related entries. This is all filled in already. And the final page that's going to be shown here is the validation reports page. I just want to say that the validation report you get at the end of this stage is not the validation report that you should be providing with your publication to journals. This is a preliminary validation report that lets you assess that the data you've uploaded is correct. After the process of annotation, you will get the full validation report, and that's what you provide with your publication for reviewers to assess your work. So that's just a quick whistle-stop tour of the deposition interface and what a deposition looks like. Now we're going to go and talk about this MMSIF file format that I've been talking to you about quite a lot, because this is what allows you to basically fill this deposition interface in automatically, and is something worth considering if you know you're going to have to do multiple depositions. So just to highlight that the PDB file format that many of you may know is really kind of considered a legacy format at this stage. MMSIF files replace this. And MMSIF files are powerful because they're both model files and metadata files, but also they can contain that metadata in the absence of a model. So you can have all of the EM metadata in one of these files, and you can provide that during upload. And that's what I did in that example there, because I was creating a deposition where I was not providing a coordinate model. In terms of how to learn more about these MMSIF files, one place you can start is the MMSIF dictionary website, which can be accessed at this link down here. This will be there for the next few slides. This is a great place to go for a full overview of the MMSIF file format and to get a full understanding of it. However, if you've never looked at an MMSIF file before, this might be a little bit challenging. So I'd recommend first going and finding an MMSIF file and actually just looking at it. And you can do that in a number of ways. You can go to the PDB websites and download one of their MMSIF files, because they're serving all their coordinates now in this MMSIF file format. But if you're just interested in making EM metadata files, you can also go to the EMDB website, and you can go to any entry from the last month or two. We recently started adding these towards the beginning of this year. And you can go to the links tab on that entry page. And then under metadata, one of these items is a sif.gz file, which contains all the experimental metadata in the SIF format. You can download this, extract this, and then just open it in a text editor. So one of the great things about MMSIF files is they're designed to be completely human readable. And when you open them in a text editor and you start scrolling through them, you'll probably quite quickly see the type of metadata that you could extract. And just to show you a couple of examples of what that looks like, I've just got two examples here. So on the left-hand side, I've got the EM imaging category, and you'll probably be able to quickly see what type of data this contains. So microscope model, FEI Titan Cryos, accelerating voltage, minimum and maximum defocus of the particles that went into your reconstruction, so on and so forth. On the right-hand side here, I've got an example of a looped category. This is arranged if you've looked at Rely on Star files in exactly the same way as that. So this is all about funding information, and you've got the column titles all up here, and then each of your columns here. So you can have either a looped category or just a single category like this. And you can probably quickly see now that you could just lift some of this information into a new text file and save that text file as a .sif file, and then that's just the file that you can provide during deposition. If you wanted to change any of these values, though, some of them are easy to change, things like defocus min and max, just so you know this is in nanometers, which is why it's so high. If you're say 1,000 to 5,000, you can change that easily. Some of these though, for example, microscope model, maybe used a Polara and you're not sure exactly what the name is that was used. This is where this website comes in useful. So every single item and every single category, so where a category is EM imaging and an item is EM imaging microscope model, has its own page, and on that page, everything is fully described. So for microscope model, you could just type that into the search and you would get this page that describes the microscope model completely. And you'd find that it has a controlled vocabulary, and you know you've used a Polara, and you can quickly see that the name used for Polara in the dictionary is FBI Polara 300. So you just put this in here, and then that would be accepted by the deposition interface, no problem. And one of these pages is this for literally every single item that we accept. So this is really the resource that you should be using. And we are very aware that publications are expecting more and more structures per publication. We have the statistics to show that. So we know that you're doing more and more entries per deposition. So you're going to have common metadata between those entries. So I do recommend making sort of a base metadata MMSIF that you can provide during deposition to all of these. And if you are providing coordinate models, that's no problem either. The MMSIF file that's outputted by software like Phoenix, for example, you can just append all of the metadata that you hold in a file to that MMSIF file, and then it will automatically be scraped. The deposition interface will recognize what is your model and what is the metadata. So that's everything I want to say on MMSIF files. When you've uploaded all of this, you've filled out the web form, which is the deposition interface. You're going to submit your deposition, and then it will go through to annotation. And I'm not going to go through this in any great detail, just to say it goes through a series of modules, and it's handled by an annotator on the side of the WWPDB. You can probably quite easily imagine what some of these do. LigMod is the annotation of ligands, SeqMod the annotation of the sequences, so on and so forth. And at the end of this, you'll get some output files, and this will include actually an MMSIF file, which has been updated with all of these annotations, and you'll get a WWPDB validation report. And this is the validation report that you're going to submit with your publication so that reviewers can assess your structure. And just to give you a couple of examples of what's in there, you've got your overall quality at a glance. This actually already has some sliders. So these are model validation metrics like clash score, Ramachandran outliers, things like this. This is what we're hoping to add to with those EM sliders that I presented before. But we already have a lot of EM metrics as well. So just some examples: orthogonal projections, Q-score maps of the coordinate model, your FSC curve. Again, there's a whole bunch of other stuff in there as well if you want to go and look at that. So that really, I think, covers everything I need to cover in terms of the EMDB and PDB deposition. Now I'm going to go on to look at a little bit at EMPIAR deposition, and this is far less structured than EMDB and PDB deposition, but it kind of means there's fewer steps and it is a bit of an easier system. So I'm going to very quickly go through this just to give you a step-by-step overview of what you do and actually how easy it is. Literally register as a new user, sign in, create a deposition, complete the deposition overview page. This is just a web form, but I'll show it to you in a second. Just on point number 4, I just want to highlight that you can grant rights or transfer the ownership of your entry to someone else. This is particularly useful if you're a PhD student or postdoc and you're leaving a lab and you need to transfer ownership to your PI. Data upload, Globus and Aspera, the two recommended methods. I'll talk to you a bit more about that in a minute. Finally, you'll complete your associate image sets, which are essentially just small packets of metadata, nothing like the amount of metadata you have to provide to the EMDB. And then you link the data that you've uploaded to this metadata and you submit your entry. In terms of EMPIAR policies, I just want to mention that all of the things that I've talked about today, WWPDB, EMDB, and EMPIAR, all have policies pages. WWPDB and EMDB, quite long pages, a lot to read there. EMPIAR deposition policies, quite concise, reads a little bit like an instruction manual. I would recommend going and having a look at these pages if you are thinking of depositing to EMPIAR. The main thing to highlight is the policies page includes information on accepted file types. Sorry, I've just realized, let me change that to a laser pointer. Here we go. Contains information on accepted file types. If the file types that are there does not include the file you want to submit, don't worry too much. You can contact the EMPIAR team. Essentially, the rule is as long as they can open the file and check it's not malicious, they're probably going to be okay with you depositing it. And just to highlight the various methodologies that are now accepted by EMPIAR, it started as a host for EMDB raw data. and it's since expanded to all of these sub-methodologies. So there really is a huge amount of data that can be deposited there nowadays. In terms of actually depositing, you'll start just by going to the EMPIAR deposition page. If you have an interest in volume EM, there's annotation of segmentations and the specimen preparation widget that might be worth looking into. For the purposes of today's talk, we're just interested in the EMPIAR deposition system. You'll start a deposition and you'll get to the deposition overview page. This is just a web form that you're going to fill in. If you get stuck anywhere, there is a deposition manual. If you're still stuck, there is a help desk that's staffed, and the staff are really helpful and will spend a lot of time trying to help you. So if you do get stuck at any point, please don't hesitate to just email and ask. In terms of file upload, Aspera and Globus, like I said, are recommended. Please use these if you can, because they're very good at dealing with uploads getting stuck halfway through and things like this and having to restart. And when you're depositing terabytes worth of data over the internet, this is something that's worth having. We can support FTP if you really need to, but it is not as resilient to losing connection and having to restart things. So we don't necessarily recommend it. And that's really all there is to say on the side of EMPIAR deposition. So just to give a quick overview, start by gathering information, make sure that it accepts the type of data you're trying to deposit. And if for any reason they don't, just contact the team and they'll get that added in for you, assuming that it's reasonable to do so. Organize your data. Each data type must be in its own directory, and also please subdivide directories possessing more than 10,000 files. This is just a file structure thing. File systems slow down quite a lot if directories contain more than 10,000 files. Set up your data transfer programs. I do recommend doing this in advance because these are peer-to-peer transfer programs, and depending on where you are, some institutions may automatically block these. And if they do, you might just need to ask your IT department for an exception, and then just deposit your data and metadata and you're all right. So that's really an overview of EMPIAR deposition. So just to summarize what we've gone through here, EMDB and PDB deposition goes through the OneDep system. Please log in with your ORCID ID, because that's going to keep all of your deposition IDs in one place. MMSIF files are also metadata files, and I do recommend looking into this if you're going to be depositing a lot of entries at once. And finally, the EMPIAR deposition system occurs through this separate system, but it is really relatively straightforward. So that's everything I had to talk to you about today, really. Thank you all for listening. I'm just going to leave this slide up just at the end here with a few QR codes if you are interested in doing any of these. So if you want to just have a quick look at the EMDB website, this QR code should take you there. The AlphaFold course that I mentioned earlier in the talk, that's accessible through this link or this QR code here. If you do want to attempt a deposition and you haven't got data ready yet, we have something called the OneDep validation server. And this is essentially a mirror of the actual OneDep deposition system, except none of the data will ever go public and you don't have to provide quite as much data. All it's going to do is produce a preliminary PDF validation report for you. And this is just quite good if you just want to have a go, but you can actually download public data that's already accessible and just put this through the validation server. It's never going to go public. You don't need to worry about anything like that. You'll know you're in the right place because if you're on the actual deposition system, it's a murky green. If you're on the validation server, it's a murky yellow. So just be aware of that. And also, the EMDB website does have an API that lets you basically get all of the entries from the website and get all of their metadata and do various things with it. I've developed a Google Colab notebook with some classes on how to do this and some instructions, and you can just run through this on your phone if you want to, and you'll get some interesting statistics out at the end. And this is just designed as sort of a teaching, both as a teaching tool, but also the classes that I've used can quite easily be just shifted into your own code and called as required to just send a request to the EMDB website and get this type of information. So with that, thank you for listening, and I'm happy to take any questions.
Okay, I'll open up the first one. So Jack, how large are data sets now? You're saying that you're 3 on to 4 petabytes soon. And now that people are collecting larger and larger data sets, do you know for EMPIAR in particular, how long an average cryo-EM data set will take to download?
Oh, I don't know how long it will take to download. I think that will depend on your geographic location and your internet connection. It's worth noting I guess you guys are in the US, so you'd use EMPIAR. If you're in Asia, PDBJ does mirror EMPIAR with the idea that these data sets are so large that you may want to something geographically more local to you to download from to try and increase those network speeds. It can take quite a long time. I think the largest data set is in over 100 terabytes, if I'm remembering that correctly. So it is in that range. In terms of the average size of data sets, I don't actually think it's going up that quickly. I think it is remaining relatively steady, but there are some outliers in that for sure. Yeah.
Do you find with the advent of a lot of new machine learning AI tools that people are now trying to download the primary data, or has it been always the case that people have been voracious downloaders of data?
I think so. There's quite a few tools that definitely have made use of Empire datasets and the EMDB in general as well. Things like Deep Enhancer, I think, use a lot of the half maps and primary maps. Priolo as well, particle picking, made use of a lot of Empire datasets and things like this. I don't think they need to use the entire archive. They will select what works for them, and I guess that's something you have to be quite cautious of when you're doing machine learning, is making sure the data's good. Otherwise, yeah, junk in, junk out sort of situation. And that's something we're hoping to improve on as well in terms of our validation metrics, to be able to label entries with quality scores. But yeah, generally there are a lot of people doing a lot of downloading of data. One of the big ones that's actually been a challenge in terms of hosting a website, though, I have to say, is the advent of LLMs, because they are scraping the entire internet. And occasionally this essentially results in a DDoS attack against the EMDB. So we have gone down a couple of times because of LLMs trying to scrape the EMDB and learn about the EMDB, but actually getting stuck and taking the entire website down. So interestingly, that one has been more of a challenge than actually people downloading lots of data.
I have to walk a bit. Give me a second.
So sorry, this is a very uninformed question, but so since people are using these AI sharpening tools like EMReady or DPM Enhancer, is it ever acceptable to use these to help with model building? And if you do, is there any? What's the process for reporting these sharpened maps?
So I'm not sure I'm an authority on whether or not this is okay to do for model building. I think people do it, and it's one of those things that if it helps you build your model in the first instance, I think that's okay and generally considered to be okay. But when you then validate that model and you run the map model validation, you should run it against the original unenhanced map. And that's what you should get your validation metrics against. And I think that's probably the most the most responsible way to go about this. We do accept deep enhanced maps into the EMDB. The way our policies and procedures work is the primary map you can really do anything you want with, but you also have to deposit the half maps. And we ask that the half maps have not been altered since they were output from the 3D refinement protocol. So the idea is that people can then recreate your primary map without those extra enhancements if they want to. But the thing that you're showing is the thing that you have built that model into.
I see, thank you.
So Jack, you said you're not the police and you're more of an archive. And given that standards are coming from the community, what has the EMDB and Empire done in terms of challenges or helping build community consensus?
So probably the most recent thing, it took a while to get published, but recently we published the white paper that came out of the validation workshop that occurred in 2020. Unfortunately, it took quite a long time to write up for various reasons, but that went out a couple of months ago. And in there are a whole bunch of recommendations that the EMDB have been working on in the background. So one example is we have a three-tiered approach to incorporating new validation metrics. So we have different web pages for different validation metrics, and while things are experimental and we're doing whole archive-wide analysis, we keep them a little bit hidden, but you can still find them. And then as these things become more and more sort of complete, we move them through to the WWPB validation reports. So yeah, it's really about engaging with the community and making sure we are following those sort of guidelines and what the community is asking for. And one of those things as well is those sliders that I was talking about, because I think in general, there just are now such an overwhelming number of possible validation metrics that you can run against your data. And actually, that's quite hard to follow, even for those that have been in the field for quite a long time, I think, let alone people first coming to the field fresh.
Can you hear me?
Oh, I can hear you now. Yeah.
Okay, sorry. I think my whole question was lost. Do you foresee any way, or is there currently an effort to try to reproduce processing pathways, like sharing star files, sharing 3D classification results, or settings, things like that, so people could go from raw micrograph to get the final map that's used for modeling when maybe you have 10 different models from a single data set?
So that's an interesting question because it's come up internally quite a few times. And I think CCPM are currently working on this type of thing with the Doppio package, where you can output this pipeliner file, which records every single step that was done. And I think you can do something similar with Scipion. But when we were really discussing it internally, the only way to properly do this would be to allow someone to deposit a container which contained all of the exact versions of their files, the exact versions of the software they use, all of this types of information. And actually, it becomes quite a complex operation to do. And whether or not this is something which is actually really useful to the wider community, we're not entirely certain. And instead, it's more about taking the data we have and using that data to present things in ways that we can only do because we have all of that data. So one example is in EMPIAR now, because we have all this volume EM data and we have all these subtomogram averages in the EMDB. And the bioimage archive is also involved in collecting a lot of CLEM data and things like this. We're looking at ways that maybe we can bring all of that data together and present it all at once. And then you might imagine that you could, search for everything in the archive which has ribosomes in tomograms, and then you could look at the positioning of those ribosomes relative to certain organelles, because you might have segmentations as well. And then you might have CLEM data that highlights other things. And by bringing all of these archives together in one pathway and then visualizing that to end users, we think that type of thing is probably more useful and something we should spend our time on. Whereas these kind of having these pipelines, we can absolutely accept those pipeline files that CCPM is working on, but us ourselves reproducing that data, I'm not sure that's something we're looking at right now. Yeah.
Thank you.
I guess for Jack, since he can see the room, how many of you have deposited anything in one depth? Can you raise your hand? Okay, that's about 3. Okay. How many people have downloaded something from EMPIAR? Okay, that's over half. And how many people have downloaded something from the EMDB? Okay, about a third. And how many people have downloaded something from the PDB? Everyone. So that means you're doing well, Jack. Any further questions?
Well, it's good to see.
Okay, well, then let's all thank Jack. Hopefully you can hear the applause or heard the applause, but thank you so much.